Hey everyone, and welcome to The Explainer. Today, we are serving up a microbial tasting menu. We're going to explore the absolutely fascinating hidden world of microscopic biotechnology that actually makes our favorite foods and drinks possible. You know, when we look down at our dinner plates, we rarely think about the incredibly complex microscopic ecosystems that created the flavors we love. But today, we're going to change that.
So when you hear the word biotechnology, your mind probably jumps straight to modern laboratories, test tubes, maybe genetic sequencing, right? But this quote hits the nail on the head. Biotechnology is definitely not just a modern invention. Everyday staples like cheese, bread, wine, and beer, those are actually some of the original biotechnologies. They're literally as old as human civilization itself. Our ancestors were out there harnessing the power of microbes long before they even knew what a microbe actually was.
Here's the tasting menu we're exploring today. First up, microbes on the menu. Then, brewing ancient Egyptian beer, the secret of chocolate, cultivating commercial mushrooms, and we'll wrap it up with the chemistry of cheese.
All right, let's dive into Course 1, brewing ancient Egyptian beer.
Now, back in Egypt's New Kingdom, we're talking roughly 1550 to 1070 BC. Beer wasn't just a casual drink to unwind with after a long day. It was a massive dietary staple. In fact, it was so vital it was literally used as a currency in their cashless society. For the longest time, historians had to rely mostly on art, like little ancient models of servants making bread and brewing, to guess how they pulled it off. But recently, researchers decided to look closer, way closer. Using scanning electron microscopy, they analyzed actual 3,000-year-old crusty beer residues scraped from ancient pots at the workmen's village at Amarna. By examining the microstructure of the ancient starch granules, yeast, and bacteria, they completely unlocked the brewing secrets of the ancients.
That microscopic analysis revealed a surprisingly sophisticated process. First, they malted the grain. Then, they cooked a totally separate batch of unsprouted grain, or sometimes malt, in plenty of water. Next up, they mixed the uncooked malt with that well-cooked grain. The enzymes from the uncooked malt went to work breaking down the cooked starches into simple sugars. Finally, they tossed in yeast and lactic acid bacteria to convert those sugars into alcohol. Pretty ingenious, right?
And the coolest part, this wasn't just some dry academic exercise. In the spring of 1996, Scottish and Newcastle breweries actually put this archaeological recipe to the test. They grew modern emmer wheat, which is the exact type of grain the ancients used, and they followed that 3,000-year-old model reconstructed from the microscopy. They produced a limited run of 1,000 bottles and called it Tutankhamun All Ale, and let me tell you, it was a massive hit. It went on sale at Harrods in the summer and completely sold out in just 3 weeks. So yeah, it definitely proves this ancient biotech produced a highly palatable and super popular drink.
Moving right along to Course 2, the secret of chocolates. It is honestly hard to believe, but raw, unfermented cocoa beans taste absolutely nothing like chocolate. If you were to cut open a pod from the cacao tree, you wouldn't find anything remotely chocolatey. Instead, you'd find seeds covered in this white, slimy, mucilaginous pulp that's packed with about 14% sugar and 1.5% pectin. You can't just roast these raw beans and call it a day. To actually get that iconic chocolate flavor, these beans have to undergo a really complex natural fermentation process that lasts up to 7 days.
This timeline reveals an absolutely incredible microbial tag team in action. Check this out. On day 1, yeasts completely dominate that sweet pulp, producing ethanol and breaking down the pectin. But the environment changes fast. By days 2 and 3, lactic acid bacteria step into the ring, eclipsing the yeasts and pumping out lactic acid. Then, as the pulp breaks down and oxygen manages to get in, days 4 through 7 are completely taken over by acetic acid bacteria. They sweep in and convert the sugars and ethanol into acetic acid. It is just a perfectly choreographed succession of microscopic life.
And here is exactly why that final stage is an absolute game changer. The exothermic reactions from those acetic acid bacteria actually raise the temperature of the fermenting mass to a scorching 50 degrees Celsius. This intense heat, combined with the acid, literally kills the bean. But in death, something kind of beautiful happens. It triggers the complex physical and organic chemistry inside the bean known as curing. Without this microscopic heat wave, you simply do not get chocolate flavor.
All right, Course 3, cultivating commercial mushrooms.
Now, this is a really unique sector of food biotechnology. Why? Because in this case, we're actually eating the fruiting body of the microorganism itself. It is an incredibly efficient way to turn low-value agricultural waste into high-value, super nutritious food. And the sheer variety we cultivate nowadays is stunning.
Obviously, the white button mushroom is the most common, but we are increasingly cultivating exotic species too, like the vibrant yellow oyster mushroom and the widely grown standard oyster mushroom. These organisms are absolute masters at breaking down materials. White buttons thrive on composted straw and animal manure, while oyster mushrooms happily grow on pasteurized wetted straw, and shiitake mushrooms grow right on wood or sawdust. And to put this into perspective, the scale of this modern biotechnology is just staggering. In the UK alone, the white button mushroom is the single highest value crop of all fruit and vegetables. We're talking a farm gate value of roughly £165 million. Worldwide, the cultivated mushroom industry is worth billions. It's a massive, highly mechanized global operation.
But nature still holds a few secrets that our biotech hasn't quite cracked yet. While we've totally mastered the white button, certain recalcitrant species absolutely refuse to be commercially cultivated. The Japanese matsutake mushroom, for example, is basically biologically intractable so far. Because it has to be wild foraged, it commands an astronomical price of around £500 per kilogram. No way, right? It's a pretty stark reminder of how valuable understanding these microbial ecosystems truly is.
On to Course 4, the chemistry of cheese.
Cheese is a product of pure, brilliant chemistry that dates back thousands of years. It likely started with nomadic tribes in the eastern Mediterranean who were just storing milk in natural sacks made from animal stomachs, completely inadvertently triggering the very first cheese fermentations. To transform liquid, short-term milk into solid, long-lasting cheese, you need two distinct biochemical processes happening side by side. First up, acid fermentation. Here, lactic acid bacteria convert the milk sugar lactose into lactic acid. Simultaneously, you have protein clotting. An added enzyme called chymosin specifically cleaves a protein called K-casein. This destabilizes the milk, causing it to coagulate and form a structural gel. Together, these two processes form the basic cheese curd.
But here's the thing. A fresh curd is firm, rubbery, and incredibly bland. To get a rich, flavorful cheddar, the cheese has to mature through a process called proteolysis. Simply put, this is the pivotal enzymic breakdown of milk proteins into smaller peptides and free amino acids during the storage period. It's this specific, microscopic breakdown that fundamentally alters the cheese, converting it from a rubbery, boring block into the crumbly, highly flavorful product we all devour. What's really amazing is the commercial application of all this chemistry. See, cheese maturation takes a long time, sometimes 6 months or more, and that ties up a ton of capital for manufacturers. So naturally, there's a huge drive to accelerate the ripening process. Traditionally, cheddar is ripened at about 6 to 8 degrees Celsius, but scientists figured out that by simply bumping the storage temperature up to 13 degrees Celsius, you can increase the rate of those proteolysis reactions and literally cut the ripening time in half. It just goes to show how a tiny shift in temperature makes a massive economic difference.
And finally, let's look at Section 5. What's next for food biotechnology?
We've seen how our ancestors harnessed microbes and how we've scaled those methods up today. But science is now stepping in to make these natural processes even more consistent and efficient. For instance, researchers are looking at adding an enzyme called pectinase directly to cocoa fermentations. This reduces the viscosity, speeds up the whole process to just 4 days, and creates a much better quality chocolate. In the mushroom industry, they're actually using genetic engineering to develop new, higher-yielding hybrid strains. And over in cheese making, the UK has widely adopted GMO-derived chymosin. This didn't just provide a highly consistent enzyme supply, it actually replaced the traditional enzymes sourced from calf stomachs, making the cheese perfectly suitable for vegetarians, which is awesome.
It really makes you wonder, doesn't it? We have leveraged microscopic life to build absolute empires of bread, beer, chocolate, and cheese over thousands of years, and now, armed with modern molecular biology, we are learning to perfectly tune these microscopic ecosystems. So the question I want to leave you with today is this. What everyday food will biotechnology revolutionize next? Thank you so much for exploring this microbial tasting menu with me on this explainer. Until next time, keep questioning what's on your plate.